Personalized cancer vaccine therapy has developed tremendously in recent years. The first step of a successful immunization is the activation of antigen-presenting cells, specifically dendritic cells, that can launch a broader immune response against a tumor. This activation not only requires a tumor-specific antigen, but also an adjuvant, such as CpG, which acts as a danger signal in dendritic cells by binding to Toll-like receptor 9, or TLR9. The fully activated dendritic cells then present the antigen to T cells in the immune system, which attack the cancer cells, producing the antigen. The presentation of multiple CpG oligonucleotides enhances the activation of the TLR9 receptor and the subsequent signaling events in dendritic cells. While many copies of CpG can be delivered to dendritic cells using nanoparticles, very little is known about if and how specific spatial arrangements of CpG oligonucleotides affect the tumor cell-directed immune response. William Xi's group at the Wyss Institute, Harvard Medical School, and the Dana-Farber Cancer Institute studies the spatial arrangements of adjuvants using square block DNA origamis in their DoriVac platform. Square block DNA origamis are arrays of DNA strands folded into a stable, three-dimensional shape. The programmable folding and stabilization is enabled by long scaffold DNA strands that are bridged and held in place by short complementary staple strands. The square block serves as a nanoparticle that simultaneously delivers both the CpG adjuvant and the tumor antigen. While tumor antigens are conjugated to one side of the square block, CpG molecules can be incorporated in specific patterns on the opposite surface. To study different CpG arrangements, the researchers vary the nanoscale arrangement of CpG docking sites with different spacing configurations. They first tested their series of DoriVac vaccines in vitro using cultured immune cells. Dendritic cells in the culture take up the DNA origami tumor vaccines by endocytosis. Inside endosome vesicles, the presented CpG oligonucleotides bind to multiple TLR9 receptors. This not only stimulates individual TLR9 receptors, but also clusters them, which stimulates the cascade of events that activates dendritic cells, and facilitates the presentation of the tumor antigens to both CD8 and CD4 T cells in the culture. The stimulated CD8 T cells are then isolated and co-cultured with target tumor cells. By measuring the activation levels of dendritic cells and T cells, and the numbers of killed tumor cells, the researchers have identified the optimal spacing configuration for CpG oligonucleotides to induce the most effective tumor cell-directed immune response. The researchers then investigated the DoriVac vaccines in vivo in mouse, melanoma, and lymphoma models. After the animals received tumor-seeding cancer cells, the vaccines were injected and tested as a cancer immunotherapy treatment. In both models, the DNA origami vaccines successfully inhibited tumor growth and prolonged the overall survival of the animals. Again, the optimal CpG oligonucleotide spacing configuration identified in the in vitro experiment demonstrated the strongest immune-boosting therapeutic effects in mouse, melanoma, and lymphoma models. The best-performing DoriVac vaccine also strongly synergized with another FDA-approved immunotherapy, eradicating the majority of the tumors in both models. The DoriVac platform is an ideal tool for investigating configurations of adjuvants and tumor antigens at the nanoscale. In the future, they could be created with antigens obtained from tumors of individual patients to form personalized cancer vaccines, and translated further toward the clinic. In addition to cancer treatment, DoriVac also has the potential as a therapy for infectious disease.